Hello students, with me Miss Yessica. Last week we have learned about the structures of biosphere, ecosystems, and biodiversity. Today we are going to learn more about that, and without further ado, let's begin our lesson. The first one is the Simpson's Index of Diversity. Simpson's Diversity Index is a measure of diversity which takes in account the number of the species present as well as the relative abundance of each species. When you have collected information about the abundance of the species in the area you're studying, you can use your result to calculate a value for the species diversity in that area. You can do this using the Simpson Index of Diversity, or you can insert D. Okay, and then for the formula, you can look at the slide. Okay, so N is the total number of the organisms of a particular species, and the capital N is the total number of the organisms of all species. So the values of D range from 0 to 1. A value near 0 represents a very low species diversity. A value near 1 represents a very high species diversity. One advantage of this method is that you do not need to identify all or even any of the organisms present to the level of the species. The higher the number we get for DT, the greater the diversity. Diversity depends on the number of different species there are, and also the abundance of each of those species. You can probably see that the diversity depends on the number of the different species there are, and also the abundance of each of those species. A community with 10 species, but where only one species is present in large number, and the other 9 are very rare, is less diverse than one with the same number of the species, but where several different species have a similar abundance. So let's take a look at the example. Okay, the question is, a sample was made of the animals living on 2 rocky shores. 10 quadrants were placed on each shore, and the number of the animals of each species in each quadrant was counted. The results are shown in the table. In the table, you can see that the painted top shells are 24 in the shore A, 51 in the shore B. Okay, you can take a look at the numbers on the table, and I don't need to mention each of them. Okay, and then we go to the number 2. To determine the Simpson index for the shore A, calculate the lowercase n divided by the capital N for each species. Square its value, add them up, and subtract from 1. Okay, repeat the procedure for the shore B. So for the table, okay, and the lowercase n divided by the capital N, you got the number 0.022. How do you get a number? You can see that for the painted top cells, the lowercase n number is 24. And then the total number of individuals for the shore A is 1,107. So how do you get the number for 0.022? You just divided 24 to 1,107. So we got 0.022. So it will repeat it for the next species, and then after that, after you got the value, you square each of the value, and then you got the result on the next column. So after that, you add them up, and then the result for the shore A is for the add up is 0.201, and after that you subtract the result from 1. So 1 minus 0.201, and then you got the result. The Simpson index of diversity for the shore A is 0.799, and then for the shore A, you just need to repeat the procedures. Systematic sampling. Systematic sampling is a type of probability sampling method in which the distribution of species is calculated based on its physical conditions such as the altitude, soil moisture content, soil type, soil pH, exposures, or light intensity change. Random sampling is not suitable for every place that you may wish to survey. You might want to investigate how species are distributed in an area where the physical conditions such as the altitude, soil moisture content, soil type, soil pH, exposures, or light intensity change. For example, suppose you want to investigate the change at the edge of the field where it becomes very marshy. In this case, you should randomly select a starting point in the field and lay out a measuring tape in a straight line to the marshy area. Correlations. Correlations is a statistical technique that can show whether and how strongly pairs of variables are related. While doing random sampling or carrying out a belt transect, you may observe that two plant species always seem to occur together. Is there, in fact, an association between them? Or you might want to know if there is any relationship between the distributions and the abundance of its species and antibiotic factors such as the exposure to the light, temperature, soil water content, or salinity or saltiness. To decide if there is an association, you can plot scatter graphs and make a judgment by eye. Alternatively, you can calculate a correlation coefficient to assess the strength of any correlations that you suspect to exist. There are three types of associations: the positive linear correlations, non-correlations, and negative linear correlations. And you can see the shape of the graph for each correlations or associations are very different. And we are going to learn in detail what the difference between each correlations in the next slide. The strongest correlations you can have is when all points lie on a straight line. There is a linear correlations. This is a correlations coefficient of 1. If S variable 1 increases, so does variable B. The relationship is a positive correlation. If S variable A increases, variable B decreases, then the relationship is a negative correlation. A correlation coefficient of 0 means there is no correlation at all. You can calculate a correlation coefficient to determine whether there is indeed a linear relationship and also to find out the strength of that relationship. The strength means how close the points are to the straight line. Pearson's linear correlations. Pearson's correlations coefficient can only be used where you can see that there might be a linear correlations, and when you have collected quantitative data as measurements, for example, the length, the height, the depth, light intensity, mass, or counts, for example, like the number of the plant species in quadrats. The first step is to check whether the relationship between the two continuous variables appears to be linear by drawing a scatter graph. The correlation coefficient should not be calculated if the relationship is not linear. The nearer the scatter of the points is to a straight line, the higher the strength of the association between the variables. Spearman's rank correlations. Sometimes you may not have collected quantitative data but use an abundant scale, or you may not be sure if your quantitative data is normally distributed. It might also be possible that a graph of your results shows that the data is correlated but not in a linear fashion. If so, then you can calculate the Spearman rank correlation coefficient, which involves the ranking the data recorded for each variable and assessing the difference between the ranks. Why does biodiversity matter? Simply, it's for 5 reasons. First, moral and ethical reasons. We share our planet with a huge range of other organisms, and you have no right to drive them to the extinctions. Second, ecological reasons. In general, the higher the diversity of an ecosystem, the less likely it is to be unbalanced by changes in conditions or traits such as the pollutions. Third, aesthetic reasons. Wildlife is a source of income for many countries as ecotourism has increased in popularity. Fourth, social and commercial reasons. Diversity increases the resistance toward the disease in the plants. Other services, such as forests, absorb carbon dioxide and may help to reduce the effect of increases in carbon dioxide in the atmosphere. Organic waste material added to the waters is broken down by the microorganisms. Transpirations of plants contribute to the water cycle, providing us with the drinking and irrigation water. Termites and ants, along with many species of fungi and bacteria, recycle elements such as carbon, nitrogens, sulfur, and phosphorus. Controlling alien species. Alien or invasive species are those that have moved from one ecosystem to another where they were previously unknown. Some species have been introduced as a biological control agents to control the pests. Unfortunately, it then became a predator of other animals. Other alien species are also maybe the escapees or animals introduced for the sport. Alien species have a variety of effects on their new environments. As well as being successful predators with few controls, they may compete effectively with the native organisms that occupy the same niche, pushing them to the extinction. They may also introduce diseases that spread to the similar organisms that have never been exposed to the pathogens. Restoring degraded habitats. Conservation involves restoring areas that have been degraded by the human activity or by the natural catastrophes such as flood, fire, hurricane, typhoon, and earthquake. Mangrove forests are found throughout the tropics. They provide important ecosystem services. They reduce the coastal erosions by reducing the effect of the strong waves during the storms, and they act as a barrier to rising sea levels by trapping sediments. Classifications. Classifications is a method to classify organisms based on their similarities and differences. Taxonomy is the study and practice of classifications, which involves placing organisms in a series of taxonomic units or taxa. In biological classifications, these taxa form a hierarchy. Each kind of organisms is assigned to its own species, and similar species are grouped into a genus, and similar genus are grouped into a family, family into an order, order into a class, class into the phylum, and phylum into kingdoms. So the domain is at the top of these hierarchical systems. So in this slide, you can see this is the classification of African boss elephants and hibiscus plants. So at the very lower, it's the species. After that, you go to genus. So the first name of the species is the genus. After that, you go to the family, order, class, phylum, kingdom, and the top is the domain. Basically, there are three domains, okay? The first is the domain bacteria. Bacteria are prokaryotic as their cells have no nucleus. The second one is the domain archaea. Archaeans are also prokaryotic, same as the bacteria as their cells have no nucleus. But the metabolisms or the archaeans is similar to that of the bacteria, but the way in which the transcription occurs as much common in the eukaryotes. The transcription means the process of one of the process in the protein synthesis. The third one is the domain eukarya. All organisms classified into this domain have cells with nuclei and membrane-bound organelles. In this slide, you can see each picture is a sample of the 3 domains. The first one is the domain bacteria. It's the cyanobacterium, the nostoc. The domain archaea, Paracoccus variesus, and the domain eukarya, the Stentorosoli. Below the domain is the kingdoms. So in this slide, we are going to discuss about the 4 kingdoms of eukarya. Okay, so it means that we only focus on the eukarya. So for the eukarya, there are 4 kingdoms. The first one is the kingdom Protista. Any eukaryote that is not fungus, plant, or animal is classified as a protochist. The second one is the kingdom Fungi. Fungi have some similarities with plants, but none of them is able to do the photosynthesis. The third one is the kingdom Planta. Planta or plants are all multicellular. Okay, multicellular means they are more than one cells. They are all multicellular photosynthetic organisms. So it means that they can do the photosynthesis. The fourth one is the kingdom Animalia. Animalia are multicellular, the same as the planta, that are all heterotrophic, means that they cannot do the photosynthesis. So it means that to get their fruits, they need to obtain them, not making them. Viruses. Viruses are acellular. They do not have a cellular structures like bacteria and fungi. Viruses are not in the classification system as we have discussed before. This is because viruses have none of the features that we traditionally use for classifications. Indeed, there is an argument that they should not be considered to be a living organisms at all. Viruses, they do, however, have particles made of proteins and nucleic acids that are found in all cellular organisms. When they are free in the environment, they are infectious, but they have no metabolisms. When they infect the cells, they may use the biochemical machinery of the host cell to copy their nucleic acids and to make their proteins, often leading to the destruction of the host cells. The energy for this process is provided by respirations in the host cells. The taxonomic systems for classifying the viruses is based on the disease which they cause, the type of nucleic acid they contain, whether it's DNA or RNA, and whether the nucleic acid is a single-stranded or double-stranded. In cellular organisms, DNA is double-stranded and RNA is the single-stranded, but in viruses, both can be either single-stranded or double-stranded. In this slide, you can see the pictures of the viruses. So the first one, you can see this is a coronavirus structures. The corona is the RNA. They have their genetic material in the form of RNA. So that's why coronavirus is highly infected and it's hard to find the vaccines for this virus as they are easily to do the mutations. And the COVID-19 in the influenza, they are all the coronavirus. And then the second one, you can see this is the Ebola virus structures, and it actually have a different form from the coronavirus. And for the virus shape, they have various type of shapes, but I will only show 2 kind of shape in this slide. Okay, that's all for today. Thank you for your attention and goodbye.